Okay, are there any questions?
A question, general question regarding the quiz tonight. Is that on rest of chapter 2 and chapter 3 and the labs?
The quiz tonight, are you seeing the screen?
Yeah.
The quiz tonight will be on chapter 3, or excuse me, that's lab 3. Will be on lab 3 and lab 4. And it will be on chapter 3 and the end of chapter 2.
Thank you.
Okay, all right. So today we're going to be talking about lab 5, streaking for isolation and culture media. On Thursday, we'll be doing lab 6. You do need to read the unknown project constructions on Canvas for next week. We're going to be talking about the unknown project, and you need to read the instructions for the unknown project before we begin the topic. Let me see if I can find that. I think it's week 5 of the lab. Hopefully you can see, you can't see all of week 5. Let me turn week 5 on. But this part you can read. Week 5 lab, you need to read the unknown instructions, this file here. And I'll be talking about it next week. Any question about any of that? All right, if there's no questions, let's go on to lab 5. The cultivation of bacteria, culture media, inoculation, and isolation. You should do some reading before you do lab 5. Hopefully you've already done this. If not, you should do it before you start the lab. Read in your textbook page 157 to 162, and then 162 to 163. Today in the lab, we have some video clips and maybe a virtual lab. I'm going to look at that later. The primary learning objectives for this lab is upon successful completion of the lab, students should be able to understand the importance of proper specimen collection and transport to the laboratory, explain the 5 principal steps used by a microbiologist to process specimens, differentiate between the various types of culture media, and explain the purpose of streak plate culture technique, AKA streaking for colony isolation. Somebody put up a chat. Remember, I do not answer the chat. If you want to ask a question, you can ask it out loud. Define the following terms. There is a selective media, differential media, and reducing media. We've talked about one type of media. We're going to talk about 3 other types of media, actually 4 other types of media. Introduction. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. If it isn't collected appropriately, you may not get the specimen, or it may be contaminated with other cultures. And then if it isn't transported appropriately and quickly to the lab, you may not be able to get the pathogen to grow. Some microbes are sensitive to desiccation, such as drying and exposure to oxygen. That would be anaerobes. And they require special transport medium. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. There's inoculation, isolation, incubation, inspection, and identification. And the lecture just finished. So let me save that. And now we go. I'll try and post that after the lab. That's the lecture Zoom meeting. And if ever I don't have the Zoom meeting posted on my YouTube website, send me an email and ask me if I posted it. Last time somebody did send me an email, and it was submitted to, I think it was submitted to YouTube, but for some reason it took a long time for YouTube to finish processing it and then to publish it, but it is published now. All right, the first step is inoculation. That's placing specimens on an artificial culture medium to initiate growth. You then have to isolate the colonies, isolate the culture to spatially separate the microbial growth into distinct colonies. You will incubate the culture, and that's just placing the culture in an appropriate environment under conditions to facilitate growth. And usually that's putting the culture in an incubator. And I'll show you an incubator in just a little bit. Then you grow up the culture and then inspect it. Let's examine the culture for the presence of microbial growth. And then you start identifying it, performing specific tests to ID any pathogenic microbes that were isolated. These 5 steps will be described in detail. We're first going to describe the different types of culture media. You're already familiar with general purpose media. That would be like nutrient agar Na or nutrient agar with yeast extract. That's Na, or tryptic soy agar TSA. These are general purpose media that are cheap, and we use them to grow a lot of microorganisms. Okay, they supply the nutrients for many organisms to grow, but not all organisms can grow on a general purpose media. A fastidious microbe will not grow on a general purpose media. These are microbes, fastidious microbes, that have very specific nutritional requirements, and they require a enriched general purpose media, and I'm just going to call it an enriched media, in order to get them to grow. An example of an enriched media is where we add 5% sheep's red blood cells to the growth media. I think it's tryptic soy agar. And then the 5% sheep's red blood cells helps the organisms to grow. For example, Streptococcus pyogenes, which causes strep throat, will not grow on a general purpose media plate, but it can grow on an enriched media that has 5% sheep's red blood cells added to the media. And whatever the growth requirements are for Streptococcus pyogenes, red blood cell supplies those nutrients. All right, any questions between enriched general purpose media and then a general purpose media? If not, let's talk about selective media. This is a media that contains chemicals that inhibit or halt the growth of some organisms. Keep in mind, when you're using a selective media, the selection isn't necessarily perfect, meaning organisms which are selected against can grow on the plate. And what you do is you grow the organism on a selective media plate as well as a general purpose media plate. And if the organism grows much better on the general purpose media, we say it is selected against on the selective media. And then second, even those organisms which are supposed to grow on the selective media may form slightly smaller colonies, or they may grow slower on the selective media than they do on the general purpose media. I can't think of an example of that, but you might be able to find an example of a selective media is one that has 7% sodium chloride added to it. That will select against organisms that do not grow in 7% sodium chloride. There's also differential media, and this media contains chemicals that produce visible differences among different groups of organisms that grow on the media. They may have a different color, or they may take on a different characteristic. And then you can look at the colonies and see differences between two different species. The differences are unrelated to how well the organism grows on the media. And lastly, the last media we're going to talk about is a reducing media. This media promotes the growth of anaerobic bacteria because low or no oxygen conditions are required to culture anaerobes. And so a reducing media is what we use to grow anaerobic bacteria. Now you should note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. And then blood agar is both differential and a enriched media. So they can be more than one type of media. All right, here we're looking at McConkey's agar. It is selective in that if the bacteria can grow on McConkey's agar, it is generally a gram negative. So McConkey selects for gram negatives and then selects against gram positives. It is differential because we can distinguish between lactose fermenters and non-lactose fermenters. If the colonies turn purple or pinkish, it is a lactose fermenter. If the colonies stay their normal color, which is usually off-white, meaning non-pinkish or non-purplish, then it does not ferment the sugar lactose. So McConkey's is both selective and differential. Any question about any of that? Blood agar is both enriched, meaning it can, in enriched media, it can allow you to grow things that would not grow on general purpose media. And I gave you the example of Streptococcus pyogenes will grow on blood agar, but will not grow on tryptic soy agar or any other general purpose media. Blood agar is also differential in that it can allow us to differentiate cells on the pattern of their hemolytic activity. So we can see 3 different types of hemolysis on this plate. There's gamma hemolysis, and that's also called no hemolysis. where you can see the red blood cells are red right up until the growth. And that's actually Greek for gamma. There's also alpha hemolysis, and that's partial hemolysis, where the red blood cells are partially lysed. You can see that the bacteria take on, not the bacteria, the blood takes on a darker appearance. Let's scroll that over. Near the cells. So the cells are the whitish alpha, and then the dark ring around the alpha is the partial hemolysis of red blood cells. And you can notice that inside the alpha, it's darker than outside the alpha. And that's from partial hemolysis. There's also beta hemolysis, and that's complete hemolysis. The bacteria here are the white, the B, and then this clear region around the bacteria, as well as inside the beta, is beta hemolysis, where all of the red blood cells were lysed. And then you can actually see through the plate right there. The red blood cells are lysed. Now you'll note that usually, if not always, you'll see a darkening of the blood around the complete hemolysis. And that's from partial hemolysis. And that's normal to see partial hemolysis outside of the beta hemolysis. Any question about any of that? All right, this table is showing you the different types of media, a general purpose media, giving you 3 types, which these 3 should be familiar to you. The enriched media, which would be blood agar plates or BAP, and that's TSA with 5% sheep's red blood cells. It is both differential and an enriched media. It's differential in that it can detect different types of hemolysis. And then there's chocolate agar, or CAP, CAP for chocolate agar plates. It's a general and enriched media. There's McConkey's, MAC, eosine methylene blue, EMB, or hectone agar HE, and all 3 of these are selective and differential media. There's PEA, or phenyl ethyl alcohol. This is a selective media. You can use PEA to isolate gram-positive bacteria because PEA selects against gram-negatives. And then there's mannitol salt agar, which is selective and differential. It's selective because it has 7% sodium chloride. Let me think about this one. Yeah, it has 7% sodium chloride in it. So that makes it selective. Only if the bacteria can grow in 7% sodium chloride will it grow. If the bacteria can't grow in 7% sodium chloride, they'll be selected against. It's differential in that you can distinguish whether the bacteria can ferment mannitol, that sugar, mannitol. If it can, the bacteria will form a yellow halo around the colony. And if the bacteria cannot ferment the sugar mannitol, it will not ferment, it will not have a yellow halo around the colony. Let me see if I got a picture of that. I do not. Yeah, that's McConkey's. I don't have a picture of mannitol salt agar. All right, any question about the different types of media? It seems to me we've already talked about that, but maybe we haven't. Oh, yeah, we only talked about the table. All right, so read about inoculation. That's putting the bacteria on a media to grow it up. Isolation is growing up a pure culture. And so when you're getting a culture from a clinical specimen, you need to put it on the media in such a way that you can get isolated colonies to grow. And Louis Pasteur developed the concept of isolation and developing a pure culture. And we'll talk a little bit about that later in the lab. His method was actually quite laborious. He grew bacteria in liquid culture and he used limiting dilution. And if 1 tube out of 10 tubes had growth in it, he thought that you probably had the growth started from one or very few cells. And so that culture was probably pure. Like I said, that was a way to get pure cultures, but it was very labor intensive. You had to have different dilutions and then 10 tubes of each dilution. And then you only got one out of the 10 tubes to grow before you would say that that was a pure culture. Robert Koch developed a much easier method for isolating growth of a culture, and that is you streak it out on a plate and you streak it to get isolated colonies. And if you get an isolated colony, there's at least a 70% chance that that colony is pure. And then you could derive a pure culture from that. And yes, 70% isn't anywhere near what we would call a pure culture. And I'll talk a little bit more about how we can up that to 95% or higher. And that is you just streak it out on the first time to get isolated colonies. And then you can say that it's 70% pure. And then you streak out from those isolated colonies on a second plate for isolated colonies. And if you get isolated colonies there, you can say that it's at least 95% probable that that culture is a pure culture. So when you're streaking, you streak like this. Let's see, we're going like that. And then you sterilize the loop and then you do quadrant 2. Then you sterilize the loop and you do quadrant 3, etc. for quadrant 4. Theoretically, someplace along the line, you'll be streaking out bacteria in such numbers that there will be very few bacteria put down, in which case you'll get isolated colonies. It doesn't matter where you get the isolated colonies. It could be quadrant 1, quadrant 2, quadrant 3, or quadrant 4. In this case, it's in quadrant 4. And where you have an isolated colony, we can say that that isolated colony is at least 70% of a chance that it is a pure culture. Let me shrink that down. Incubation is just growing the cells in an appropriate media, usually in an incubator. And there we're looking at an incubator. This incubator is very fancy. You can control the humidity. You can control the temperature. You can also control the gas, like the levels of O2 and CO2. most of the incubators that we have in the lab only control the temperature and the humidity. And then you incubate for an appropriate time, usually overnight or a day, to get colonies. You then inspect the culture to see visible colonies. And if you don't have visible colonies, you put it in the incubator for another day and then inspect the culture. But if you see isolated colonies, then you know that you have a chance of deriving the culture of interest, meaning the pathogen that was making the patient sick. You can then start isolating the culture and identifying it by making various descriptors about the colonies, colony size, shape, margin, elevation. We'll talk to that. You can refer to Lab 01 for a review of that. You cannot identify it just from that, but you can then identify it by running tests on the bacteria. All right, so know the terms of this lab. And then do the laboratory exercises. You're going to be watching a video on the inoculation of agar plates using streak plate culture technique. You only need to watch the video to 4 minutes and 15 seconds. You should note that this streaking is only using 3 sectors. In the pictures above, we had 4 sectors. And both of them are good ways to streak out plates. Then watch a video on beta hemolysis, alpha hemolysis, and gamma hemolysis. Then watch a little video on McConkey's agar. Watch only to 3 minutes and 50 seconds, meaning the movie, you don't need to watch all of the movie. So watch it to 3 minutes and 50 seconds. I'm looking to see, I think I do. Let me look into that one. And then watch a little movie on mannitol salt agar. And you might have a virtual lab on practicing for colony streaking. Let me look and see if that's the worksheet. It's not what I wanted. Let me go back here. All right, let's go to the lab from here. I don't remember if you have a virtual lab or not, so that's what I'm doing.
Mine is crossed off.
It's crossed off?
Yeah, on my.
All right, then there is no virtual lab for today. That's what I was looking to see if there was a virtual lab. If it's crossed off, that means you need Flash to run it, and Flash is no longer running because they've turned it down, stopped working it, and we haven't found another virtual lab. Yeah, it's crossed out. All right, so then after that, do the worksheet. Describe the natural circumstances for where Staphylococcus species might benefit from salt tolerance. And please write these in the exercise, not in the lab manual, and submit only the worksheet. The worksheet is a much smaller file than the lab manual, and I don't want to my Canvas website to become full. So please submit only the worksheet. 2, hemolysis benefit a hemolytic organism. Write your answer. 3, imagine that you have culture containing a single bacterial species. It could be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB, ethylene methylene blue plates to identify the unknown bacterial species. How would you use these plates to help you identify what the species is? You have to discuss each of the medias, mannitol salt agar, blood agar, and EMB. Then answer 4, what is the main goal of streaking for isolation? And that's colony isolation. 5, what happens to the growth on the plate if you forget to sterilize the loop in between sectors when you're streaking for isolation? Be specific, discussing each sector. Note that it's not saying that you're not sterilizing the loop before you start sector 1, but you're not sterilizing the loop between sector 1 and sector 2. And so what's going to happen? And then 6, what happens to the growth on the plate if you transfer from stock culture to inoculate each of the sectors, meaning you pick up on your loop stock culture to set up sectors 1, sectors 2, sectors 3, etc. This is specific, discussing each sector. What I'm going to look in is specifically how 5 and 6 differ. So if you don't have a difference, it's not going to be correct. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species from a mixed culture. How can you use this plate to create a new pure culture of one of the bacteria? Be specific in your answer. And 8, in the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective, selective, or differential by placing an X in the appropriate column. There may be more than one X in the row. You may use the table of media in the lab module to answer these questions. Like nutrient agar is, I'll tell you the first one, it's a non-selective media only. So fill the table in for question 8. And then 9, which of these plates, this one, or that one, used the streak plate technique, meaning was inoculated using streaking for colony isolation. So A, B, or C. All right, any questions about any of this? If not, go ahead and start the lab. I will be here until 8 o'clock to help you with any questions you have. However, I might grab a bite to eat, but I'll be back. I'll be back at my computer within a few minutes, within 2 or 3 minutes. Okay? All right.
Can you just tell me what chapters you're going to be covering this on Thursday and then next week? I'm going to be traveling, so I need to know what to bring with me.
Okay, can you repeat that? Let me turn up the volume and maybe
That's
I'll
fine.
hear you better.
So what chapters are you going to be covering on Thursday and then next Tuesday and Thursday?
All right, you can take a look at the syllabus. Let me share my screen.
Thank you.
So next week, we're going to be covering chapter 5 and hopefully get started with chapter 6, a little bit into chapter 6. And in the lab, we're doing lab 7. We're also going to be discussing the unknown project and starting the unknown project.
Okay, perfect.
Okay?
Thank you.
And note, there is a quiz next week. Okay.
What day? Yeah, is that
It's,
Tuesday or
take it from 8 p.m. Tuesday night to 8 p.m. Wednesday night.
Okay, can I email you about that?
Yes.
Okay, perfect. Thank you.
All right. Stuff.